Good morning, everyone. My name is Safomuddin. I'm PhD candidate at Shanghai Institute of Materia, Medica, Chinese Academy of Science. I'm grateful to share my work today in asymmetric synthesis of Finerenone via Owing chiral auxiliary in 9th World Chemistry Conference and Exhibition. First of all, I would like to briefly introduce about our research laboratory. In our research laboratory here in Shanghai Institute of Materia Medica, Chinese Academy of Science, our laboratory led by Professor Shan Jingshan as a principal investigator and doctoral supervisor. Until now, our research team, under supervision of our professor, work in 3 major directions like viral infection, neuropsychiatry, reproductive health, and finally. And over 20 years, our team built an integration platform of 2 subsidiaries, such as research and development for antiviral drug, and secondly, production system of green synthesis process for API, research development, and manufacturing. Here you can see our achievement regarding APIs, like our new APIs and generics API. Among the mentioned APIs, today I would like to introduce about the latest update of Finerenone in our laboratory. Finerenone is a mineralocorticoid receptor antagonist, and this receptor antagonist can be classified by 2 classes of the compounds, such as steroidal mineralocorticoid receptor antagonist, like 1st generation and 2nd generation, such as spironolactone and eplerenone. For the 2nd class of the compound, we can say the 3rd generation, which we call nonsteroidal mineralocorticoid receptor antagonist. These mentioned APIs can be used for the treatment of heart failure, and currently heart failure is one major drawback among cardiovascular disorder. However, treatment of them with steroidal mineralocorticoid is quite challenging because it has several side effects, such as increasing the potassium level in the body to make hyperkalemia. However, as an alternative non-steroidal antagonist drugs, Finerenone can be used for the treatment of mentioned disease with a better profile, such as cardiorenal protection. And that's why Finerenone, as a mineralocorticoid receptor antagonist drug, was approved in 2021 by United States Food Drug Administration for the treatment of chronic kidney disease associated with diabetes type 2. Additionally, for further expanded its indication in 2025, FDA added for the treatment of heart failure with a patient who is suffering from ejection fraction of with 40% higher. And as a useful API, I think existing method for synthesizing of this challenging molecule is suffering from a lot of disadvantage, such as improving stereoselectivity or purification of stereoselectivity. However, with our team, we design new synthetics method regarding Finerenone along with its key intermediate. And now I would like firstly briefly introduce about the key intermediate of Finerenone, which we call 4-amino-2-pyridones. And regarding this key intermediate, here is a lot of synthetics method, but among of them I choose a commercial route. And also commercial route has some disadvantage, but I want to briefly introduce about it. In this synthetics method, pyridone was synthesized from chloroaminopyridine in the 4th step of the sequence, such as oxidation, nitration, and reduction in hydrolysis. Here you can see, I think, using hydrogen peroxide as the oxidant agent, it is quite dangerous, and as well as for the second step of the reactions, they use a mixture of nitric acid and sulfuric acid. And for the reduction reaction, here is as a reductive agent here use iron. For example, workup of this procedure of the reaction is quite challenging, because after doing workup, a lot of waste will produce. In general, the overall yield of this synthetic method is not quite attractive. However, it's around 20%. Regarding this challenging, we design our 1.3-step synthesis approach, starting from malonyl chloride and vinyl ester. And the advantage of our synthetic method is that during the processor, for example, after getting nucleophilic reaction between malonyl chloride and vinyl ester, we got intermediate 12 or 30 esterification to get intermediate 13. In this method, this intermediate, without any purification, further purification, was charged for the next step of the reaction. In the next step of the reaction this one is a cyclocondensation reaction. Later on, I will talk about during the mechanism of the reaction, I will talk the purpose of ammonium acetate in this reaction procedure for the simultaneous formation of carbon-nitrogen bond. Then with our designed method, then we focus to applicability of our method and to check the different substrate scope for our reaction, and from different electro-withdrawing group or electro-donating group, or checking with some temperature-sensitive functional group, we was found it all of this functional group is well tolerated for our designed method. Among this substrate, you can see here is substrate 14P. This one is the key intermediate of Finerenone. However, after getting this achievement, we shifted our attention for the large-scale synthesis of key intermediate of Finerenone. For example, here you can see we scale up our reaction to 100-gram scale, and we achieve 51% overall yield, which means more than 75% per each steps. And the purity of our compound also is quite attractive, around more than 99%. And also during this study, during this large-scale synthesis, we didn't use any further purification with chromatography, just we find optimal recrystallization to get our compound as a pure. And as well as here, we did some impurity study and impurity control. Regarding impurity control, the temperature is the best parameter to control our impurity. For example, when also previously we did optimization for temperature, and we was found that this 85 degrees Celsius is the best optimal for the controlling impurity. And also we identified our the impurity which obtained during the procedure, and we was found it and figure out with the different analytical equipment. Then, our synthetics method is a novel synthetics method. We try to design a mechanism of the reaction. For the two-step of the reaction, it is common reaction. The reaction mechanism is not challenging, but for the final step of the reaction, like for cyclocondensation reaction, We base it on our result and some published work. We designed the synthetic method, which start from ketoester sorting and amino and acid, this salt, ammonium salt, to get the first carbon-nitrogen bond formation via amine formation. And for the second step of the reaction, ammonium acetate can conduct with the intermediate A to make the complex which is connected with a donor and acceptor's bond. However, afterward, following nucleophilic substitution and intermolecular proton transferring, we achieve the intermediate D. Then, following two steps of the sequence, elimination of ethanol, we got intermediate F, and following carbon-nitrogen formation band and elimination of acetic acid, we got in the pyridone 2. And in this reaction mechanism, we was founded here is ammonium acetate can be used as for introducing of 2 nitrogen atom into this scaffold, and additionally the acetic acid which eliminate during this reaction can promote our reaction as a catalyst. And however, with the intermediate of finerenone in a hand, then we focus our attention on synthesizing asymmetric synthesis of finerenone following the retroscientific analysis which we designed. Regarding finerenone, I can say actually existing method nowadays, I mentioned it before, existing methods are suffering from a lot of disadvantage. For example, if we look for medicinal chemistry route during medicinal chemistry route or commercially route of finerenone, for purification of enantiomer of S-active finerenone, for the purifications they use chiral HPLC. And also this synthetic method which I designed here, it's published during the third phase of clinical trial of finerenone. And from stereochemistry side, we can say this synthetic method is quite attractive, like it can be some innovative here. But unfortunately, for example, regarding hydrogen transformation, but unfortunately, they use some catalysts like phosphoric acid catalyst, which is quite costly. For example, in our group, we are working on APIs from laboratory study till the industrial study. We always focus on, first of all, it should be economically available, and also in same times, it should obey the principle of green chemistry. However, in this regardless, we designed the synthesis method for finerenone starting from a chiral E1 auxiliary, and our synthetic method overall yield is quite attractive compared to published one. For example, overall yield is around 31%. It's included for the 6th step of the sequence. And here you can see our synthetic method from very beginning till getting our API. First aim of our work is that to improve the stereoselectivity of finerenone. In this regardless, we choose a different chiral auxiliary group, and we was founded among this auxiliary group, among them 4-benzyl-2-oxazolidone is the best optimal reaction because it can reach the stereoselectivity around to 86 to 14 ratio. After finding this achievement, we got this ester, for the first step of the reaction to get the ester compound 17. And for this step of the reaction, we don't need any purification by column chromatography, just via ordinary recrystallization, not only for first step first. For step first, second, and third one, we didn't use any purification, and we got by recrystallization. And for the cyclization step is a key step of the reaction, which I mentioned it. We use different chiral auxiliary to achieve the naphthyridine chorea or finerenone with high stereoselectivity. And afterward, we focus our attention on how to purify enantiomer from each other. In this regard, we use the recrystallization method to separate our enantiomer 21. Then, following orthoalkylation reaction, we introduce the ether group in the naphthyridine chorea of finerenone. For the final step of the reaction, actually this one is transamidation. We can say transamidation reaction. In this transamidation reaction, when we try the reaction in the presence of ammonia, and we observe it that impurity, except finerenone, here is some impurity can produce. However, for controlling this impurity, we did different optimization, and we was found it for controlling of this impurity if we increase the concentration of ammonia, it can reduce the amount of the impurity to 95 to get the selectivity between finerenone and impurity 23 to 95 to 5 ratio. And finally, in conclusion, what I want to say, our synthetics method, for example, let's say about the one-pot cyclocondensation for pyridones. Actually, our synthetics method is not only about synthesizing key intermediate of finerenone. Key intermediate for finerenone is the only one member of our substrates. However, previously synthetic method for pyridones in general, for two pyridones, they are all suffering for introducing of two nitrogen atom in a scaffold. However, our designed method can solve the problem which was produced with the previously synthetic method. And additionally, synthetic method for finerenone, we can say we improve the stereoselectivity of naphthyridine chorea of finerenone, and our overall yield also it's quite attractive. Thank you very much for your attention. If any question, please.
Thank you, Doctor. Thank you for your presentation. Now the session is open for queries. Any questions, please? Any questions, please?